Hello guys, welcome to this course. In this lesson, we'll be studying about mitochondria. Okay, this course is presented by me, Navneet Sharma. You can know more about me from here. You can follow me on this link. And if you like the lesson, kindly rate, review, and recommend. Beginning the lesson. First of all, I would like to tell you that I will be making two lessons on the same topic, mitochondria. Okay, so this is the part one of this topic. We'll start with the basics. Definition. Mitochondria are the center for cellular respiration. It converts chemical energy into kinetic energy. Okay. Info. In 1857, Kolliker observed mitochondria and called them as saccharosomes. Fleming called them as phyla. Altman in 1890s called them as bioplasts. Benda gave the name mitochondria. Porter and Palladay described their electron microscopic structure. Mitochondria are present in all eukaryotic cells. Okay. Now the size. The length varies from 1.5 to 10 microns. The smallest mitochondrion is seen in yeast. It is approximately 1 millimicron in length. The width is 0.5 to 0.7 microns. Which is generally found, but the oocyte of amphibian shows 20 to 40 microns length of mitochondria. Okay, it is mostly the largest size of the mitochondria. The shape, they are filamentous, or they may show rod, spherical, or thread-like structures. Number. The micromonas, in micromonas only one mitochondrion is present. In a liver cell, 1000 to 1600 mitochondria are present. The highest number of mitochondria are seen in the cell of a flight mussel. However, cell of a green plant contain less number of mitochondria. Okay, in RBCs, that is red blood corpuscles of mammals and other higher animals, mitochondria are absent. Structure of a mitochondria. The mitochondria is covered by two layers. It shows outer membrane and inner membrane. In each mitochondrion, two chambers are present. The first one being the outer chamber and second one being the inner chamber. The outer chamber, it is a continuous membrane which covers and protects the mitochondrion. It separates the mitochondrion from the cytosol. It is permeable. Inner membrane, it is projected inwards as a cristae. The membrane shows two faces. The outer face, the C face, or called as the cytosol face, and inner face is called as M face or the matrix face. On this M face, a number of knob-like structures are present. The outer chamber has the space between outer and inner membranes. It is 60 to 80 angstroms. It is filled with a fluid. The inner chamber, it is called as the mitochondrial matrix. It is filled with a gel-like substance. It contains many enzyme systems. In 1963, Ness observed the presence of DNA molecule in the mitochondria, which was a very important discovery. Okay, so you can first of all have a look at the diagram for normal mitochondria. Okay, we have the DNA present, the ribosomes are present, a matrix is present here, the outer chamber, the inner chamber, and the intermembranous space. Okay, and these are the cristae, that is the inner foldings of the wall. Okay, variations of cristae. Normally the cristae are perpendicular, but variously arrangements can be noticed. Okay, parallel cristae. In the nerve cells and the cells of striated muscles, the mitochondria will show parallel cristae. Concentric cristae. In the spermatogonia of men, the mitochondria will show concentric cristae. Haphazard arrangement. Irregular arrangement of cristae is called as the haphazard arrangement. Spherical cristae. In the spermatocytes, the cristae are spherical. This is a diagram of different kinds of arrangement of the cristae in mitochondria. Okay, you can refer to it. Next we have the mitochondria without cristae. Very rarely the inner membrane is smooth it will not show any type of cristae or reduced cristae. In the cells of opossum testes, the cristae are reduced in the mitochondria. Okay, moving on. Mitochondrial DNA. Mitochondria contain one or two molecules of DNA. Mitochondrial DNA is circular. Okay, remember that. It is a highly twisted, double-stranded molecule. Okay, it is deoxyribonucleic acid. Okay, we all know that DNA is always double-stranded. Rabino, he stated that the mitochondrial DNA contains more G and C content, that is guanine and cytosine content, than in nuclear DNA. The molecular weight of mitochondrial DNA is about 9 to 11 millions, a million microns. Okay, this DNA is the capacity of multiplication. DNA polymerase enzyme is present. This DNA will produce RNA. It is believed that it may take up the production of some proteins also. That is why it is found in the mitochondria. Okay. Next we have the functions of the mitochondria. First one is the ATP synthesis. That is the powerhouse of the cell. Okay. You must have studied in the lower classes that mitochondria is called as the powerhouse of the cell. It brings oxidation of food, hence Krebs cycle reactions, electron transport system, enzymes are located in the mitochondria. Okay, by the oxidation of food, energy is liberated in the form of ATP, that is adenosine triphosphate, in which oxidative phosphorylation takes place. Okay, next we have yolk formation. Mitochondria are responsible for the formation of yolk. There's a typing mistake. Okay, forgive it. In the developing ovum, granules are formed in the matrix. They arrange large masses. Mitochondria is converted into yolk storing body. Okay. Next we have mitochondrial sperm formation. When the spermatid becomes sperm, the mitochondria will form a spiral around the axial filament. This is called the Nabin kern. It forms the middle piece of the sperm. So this is also helpful in providing energy to the moving sperm. Mitochondrial functions. So the other functions are the origin of new system. It is believed that some of the cell organelles may originate from mitochondria, but it's just a belief. Heat production. In the oxidation of food, ATP is released. Only 45% of total energy is trapped in the form of ATP. The remaining of 35% of ATP will come out as heat. In birds and mammals, this heat is useful for the maintenance of body temperature, as we are warm-blooded animals, so we need to regulate our body temperature according to the outside environment. ATP released during respiration because of mitochondria will take part in the many biosynthetic parts of the cells. Okay, and it is necessary for maintaining the body functions. So thank you guys for watching this lesson. In the new upcoming lesson, I'll be talking more about the mitochondria. So keep updated. Thank you.